And our next speaker will be Ofer Gerlitz.
Hello. In the early embryo, the gonad is bipotential, and it can form either a testis or an ovary. As opposed to male development, very little is known about female development and the gene network regulating female gonadal differentiation. And it was, until recently, it was thought that female gonadal differentiation is a default process occurring in the absence of SRY. However, ovarian development is an active process involving multiple signaling pathways and crosstalk between somatic and germ cells. Aberration of this complicated process occurring in disorders of sex development can cause severe outcomes, including major structural anomalies, gonadal dysgenesis, problematic gender assignment, and infertility. XX ovarian dysgenesis is a rare disease resulting from defective development where the embryonic gonad is replaced by functional fibrous tissue. The associated hormonal failure also prevents the development of secondary sex characteristics, resulting in a sexually infantile female appearance and infertility. Family J is a consanguineous family affected with XX ovarian dysgenesis. A detailed clinical analysis revealed 6 female relatives, all with symptoms, classical symptoms of ovarian dysgenesis, including absence of menstruation, lack of pubertal development, abdominal ultrasound and MRI showed hypoplasia of the uterus, undetectable ovaries, blood tests showed low levels of estrogen and progesterone, and high levels of gonadotropins. We performed SNP homozygosity mapping in combination with whole exome sequencing, and to make a long story short, we identified a recessive mutation in the nucleoporin 107 as the causal candidate. This mutation segregates with the ovarian dysgenesis phenotype, was not present in available database or in 150 healthy ethnically matched controls. So we conclude that this mutation is the causal candidate for the ovarian dysgenesis in family J. However, at this stage, we still didn't have a functional proof to link NUP107 or the identified missense mutation with ovarian dysgenesis. NUP107, nucleoporin 107, is an essential component of the nuclear pore complex, and as NUP is highly conserved and mutated aspartic acid reside within a highly conserved stretch of amino acid, we decided to perform a gonadal knockdown in Drosophila to assess a possible role of NUP107 in ovaries development. So knockdown of NUP107, specifically in somatic gonadal cell using traffic gem GAL4, resulted in female sterility, whereas males were fully fertile. These sterile female laid 15-fold less eggs than the wild type. Importantly, also, when we looked at their ovariole, we saw general disintegration, DNA condensation, accompanied by caspase reactivation. Importantly, when we knocked down NUP107 in the germline using Nanus Gal4, there was no effect on fertility. Males and females were fully fertile. This suggests that the gonadal function of NUP107 is restricted to the soma and not to the germline. We then generated a comparable Drosophila animal model where we deleted the endogenous NUP107 gene located on the second chromosome and rescued it with a genomic fragment containing the NUP107 gene fused to RFP. We made two versions, the first being a wild type, and the second containing the missense mutation found in family J. We found that the mutant-rescued females had impaired fertility. Specifically, they laid twofold less eggs, of which fourfold less hatched. Overall, they had 7.2-fold less progeny. In addition, when they laid 70% of the eggs they laid were aberrant, so they showed the aberrant eggshell morphology compared to only 25 aberrant eggshell morphology in the wild-type rescue eggs. When we looked at the ovarioles, here the effect was striking. We saw 71% of the ovarioles in the mutant-rescued females showed general disintegration of the ovarioles, including DNA condensation and caspase reactivation. In contrast, in the wild-type rescue, we saw this phenotype only in 9% of the ovarioles. So we still wanted to see something similar to ovarian dysgenesis. Here we are talking about perturbation of oogenesis. So I asked the student to look whether, so the students, when they dissected the female ovaries, so they looked for ovaries. So I told them, try to look for no ovaries, because we are dealing with ovarian dysgenesis. So we were astonishing to see basically ovarian dysgenesis-like phenotype in over 30% of the mutant females. These include non-developed ovaries, you see here, and underdeveloped ovaries. Strikingly, I think, if you can appreciate, we can see in the same female fly, one normal-looking ovary, and on the other side, ovary which hasn't developed. Also here, instead of ovaries, we see a fat-like tissue. So this is really reminiscence of the ovarian dysgenesis that we see in humans. So this raised the question, at what stage of development does the NUP107 missense mutation abrogate ovarian development? So we looked at the larval gonad, and we found that the larval gonads in the mutant were present to the same extent as in the wild-type rescued larvae. This indicates that the developmental failure occurs only after the larval stage. However, we hypothesize that the genetic and molecular cause are already present at the larval stage. So consistent with this notion, we found that the larval gonad actually contains a dramatic excess of primordial germ cells. You see here, this staining with VASA. So this is not to say that this is the cause of the developmental failure. This, in our opinion, just suggesting that the developmental program is already abrogated in the larval stage. And we see the failure in later stages. We are currently looking for differences changing in major signaling pathway and the targets between the wild type and the mutant rescued larval gonads. We are using a panel of antibodies. We also just now comparing the transcriptome of the wild type and the mutant larval gonads using RNA-seq. And we are preparing to do proteomics also. So, to summarize, our result identified and confirmed NUP107 and the identified missense mutation as the genetic underlying cause for the observed ovarian dysgenesis. We generated a comparable Drosophila animal model where mutant female flies show an ovarian dysgenesis-like phenotype. And we aim to use this model to elucidate the genetic involved in ovarian development, hoping to shed light on human XX ovarian dysgenesis. Finally, I would like to thank the student who did the work, our lab technician Rachel Khalifa, two excellent students, Tikva Shor and Dina Reckler. They were assisted by Nili Fardian and Amatzia Dreyfus. And we have an ongoing collaboration with the endocrinologist David Zanken and with Professor Efrat Levi-Lard and her excellent PhD student, Ariela Weinberg. Shukron. Thank you. Okay.
Just a quick question. So you talk about the ovarian phenotype. Did you test other phenotypes with this mutation in other tissues? And then second question, so NAPs are, of course, very exciting proteins involved in heterochromatin and chromatin architecture in different developmental stages. Have you expressed or made this mutation in some cell culture where you could look at the heterochromatin or chromatin architecture? Or what do you think about that approach?
Okay, to the second question, because I don't remember the first one. So, how NUP107, which is essential nuclear pore, structural protein, can affect specifically the development of the ovaries. I also would like to mention that these poor ladies, with the ovarian dysgenesis, they are perfectly healthy, except for the ovarian dysgenesis. And when they were treated with hormones, they have menstruation, they develop sex, their uterus developed, and basically they are normal women. They even can bring children if they get a donation of eggs. So how NUP can do the job? So as you mentioned, NUP is involved, the nuclear pore is involved in regulation of transcription. But the way I envisage it is that we saw that you can see in the same female one ovarian dysgenesis, one ovary, and the one looking perfect. So this tells me that something is on the border. So there is some signaling which either you overcome the threshold or not. So I think, so it was shown recently that although NUP107 is a structural protein, when there is a DNA damage, the ARC protein, the APAF1 homolog, part of a protein which is part of the apoptosome, after DNA damage, he has an independent function where it translocates to the nucleus. And there it does what it do. But the translocation is depending on specifically on NUP107. So this is an example where you have a specific, so NUP107 may regulate the translocation of certain component which is required, especially, not only in my opinion, to ovarian development, and this is somehow by the mutation is affected, is reduced, and you either cross this threshold or not, and you can get this phenotype. Okay. Thank you.
All right.